and welcome everyone to the webinar, PIRCHE Epitope Matching in Kidney Transplantation, a Case Study. My name is Andrea Zimmerman, and I work on the North American sales team. At GenDx, we organize webinars at least once a month on various subjects, such as case studies, product updates, and scientific updates. Today, we have teamed up with our colleagues from PIRCHE for a case study webinar. Before I give the word to our guest speakers, I want to make a few practical announcements. Our webinars account for ACHI continuing ed credits, and we will send you a certificate of attendance by email. So this should be sent within the next week. Next, if you experience some technical problems during the webinar, please try to refresh your browser by pressing F5. At the end of the presentation, there will be some time for questions. So if you have any questions, please type your question into the chat box on your GoToWebinar screen. Now let me introduce to you the speakers of today. Dr. Eric Spierings is Associate Professor and Lab Director of the HLA Laboratory in the University Medical Center Utrecht in the Netherlands. His current scientific interest focuses on the role of indirect recognition of mismatched HLA in transplantation and on post-transplant immune monitoring via next-generation sequencing of T and B cell receptors and immunoglobulins. Matthias Niemann is the Director of Technology at PIRCHE in Berlin. His focus is on implementing the PIRCHE web service and transplanting the algorithm from a research prototype into a commercial tool suite. He supports research teams all around the globe, has co-authored several PIRCHE-related publications, and regularly presents scientific abstracts at international conferences. I would now like to give the word to our guest speakers for the presentation, PIRCHE Epitope Matching in Kidney Transplantation, a Case Study.
Thank you, Andrea, for the introduction, and hello, everybody. In this webinar, I will indeed focus on the role of T-cell epitope matching in solid organ transplantation, and I will start with explaining how T-cells and T-cell epitopes are involved in the antibody-mediated graft rejection. And after that, Matthias Niemann will show you how these principles can be used in the clinical context, applied to real cases. Second, I will move on into the immunological T-cell memory and the effects of the kidney transplantation thereof. And finally, Matthias again will illustrate this T-cell memory concept based upon a few cases using the PIRCHE algorithm. Before I continue, I would like to display my disclosure that is related to the topic of today. The success of solid organ transplantation is significantly determined by histocompatibility between the donor and the recipient. And this aspect is illustrated on this slide by showing you the graft survival rates that are observed in recent Eurotransplant cohorts. And it's clear that the higher mismatch combinations here in red for the zero mismatch ones and in blue for the single mismatch ones perform better in terms of graft survival compared to the mismatches with 4, 5, or 6 mismatches here in fuchsia, orange, and gray. And these effects are the result of recipient alloimmune responses that are directed towards foreign proteins, in this case mismatched HLA, on the transplanted organs. With respect to alloimmune responses in HLA, mismatched transplantations, the adaptive immune responses form the most important compound of the immune system, causing these problems of immune-mediated rejection. And the adaptive immune system involves antigen-specific responses with two major branches. One is the B cell branch down here, ending up with antibodies as effector molecules. And the other is the T cell branch with CD8 cytotoxic T cells and CD4 T helper cells as effector cells. And the cytotoxic T cells recognize T cell epitopes presented in the context of HLA class 1, whereas the T helper cells recognize epitopes presented on HLA class 2 molecules. In the clinical context of kidney transplantation, there's a renal allograft here that delivers proteins to dendritic cells, and these dendritic cells can then present those foreign proteins to B and to T cells, leading to antibody formation, CD4 cells, and CD8 cells. And we call this phase the sensitization phase. After that, the T cells and antibodies can have their effect on the transplanted organ, causing antibody-mediated rejection or T cell-mediated rejection. The CD4 positive T helper cells play a central role in the sensitization phase by stimulating both the T cell mediated pathway and the antibody mediated pathway. And these T helper cells do so by stimulating the CD8 positive T cell differentiation and also the B cell differentiation. So schematically, we have the two effector mechanisms here on the left, attacking the organ with the antibodies and the cytotoxic T cells, causing the damage. And we have the T helper cell that plays this central role in both of these branches. And today, I will particularly focus on the interaction that these T helper cells have with the B cells and how they act and what that means for evaluating HLA mismatch. So why is this help to the B cells so important, and where does it exactly lead to? Well, actually, this help triggers three important aspects leading to finally producing effective antibodies. And the first one is isotype switching, where the constant domain of the immunoglobulin can change from, for instance, IgM to IgG. And this process of isotype switching leads to a different effector mechanism, as the constant domain of the immunoglobulin is responsible for that. The second effect is the creation of memory. And memory is important to more rapidly evoke an immune response upon a second encounter of the antigen. So a first exposure will lead to a low IgG response, here in green, and a subsequent exposure will rapidly lead to a very high IgG production. And this effect is well known, for instance, in the context of vaccination. And finally, the T cell help to B cells triggers the process of affinity maturation of the antibodies. The B cell is able to change its sequence of the variable domain in the immunoglobulin in such a way that the affinity of the antigen with the antibody can be enhanced. The next question then is, of course, how the T cell provides this help to the B cell. How does that work? What are the interactions and requirements? Well, functionally, the T cell needs to provide signals to the B cell via soluble factors as cytokines or via membrane-bound receptors and ligands to provide co-stimulation. And an important trigger in these processes is the cognate interaction of the T cell receptor, here in green, with an epitope in the context of HLA class 2. And the class 2 antigens present these T cell epitopes via the peptide binding domains of the HLA molecules. These T cell epitopes are derived from internalized antigens that are being processed into smaller fragments. For internalization of the antigen, the B cell receptor is of importance, and it's crucial to understand that this B cell receptor has the same specificity as the antigen it's going to produce. They are products of the same gene segments. Thus, a B cell that will produce, for instance, HLA-A2 antibodies has a B cell receptor that binds HLA-A2. And therefore, a successful antigen requires both a B cell epitope and a T cell epitope. And this latter concept is called the principle of linked recognition. So the generation of a mature antibody response requires the presence of both an antibody B cell epitope and an HLA class 2 restricted T cell epitope within the same antigen. And this phenomenon is called the linked recognition phenomenon. For HLA antibodies, the linked recognition means that an immunogenic HLA antigen must also contain a T cell epitope. The PIRCHE algorithm is a computational tool that addresses the presence of these HLA class 2 presented T cell epitopes in mismatched HLA. So After 2 proof of principle studies in kidney transplantation and pregnancy, respectively, we evaluated the PIRCHE2 associations with HLA antibody development in a large kidney cohort. And this first large retrospective study was executed in collaboration with Niels Lachmann and Constanze Schönemann from the Charité in Berlin in Germany. And the results that you see here show that the recipient donor pairs with higher PIRCHE scores, for instance here in red, more than 90, had a significantly higher risk for developing de novo donor-specific HLA antibodies than the pairs with a low PIRCHE2 score, here in black. The data also suggested that this correlation is likely not a linear one, but a stochastic one. So in a subsequent study on kidney graft failure, we therefore converted the PIRCHE2 scores by calculating the natural logarithm of it and evaluated these converted scores as a continuous parameter. And these studies were done within the ProCare Consortium, a collaborative effort including all Dutch HLA laboratories and kidney transplant clinics, and is funded by the Dutch Kidney Foundation. And the results of the analysis show that of all HLA factors, the natural log of the PIRCHE2 score was the strongest associated factor, resulting in a hazard ratio of 1.11 per natural logarithm of PIRCHE2, shown here. So following these first studies, PIRCHE2 effects have been observed in many other cohorts of solid organ transplant pairs, including also liver, heart, and pancreas transplantation. And given the time restrictions, I refer to these publications and to the PIRCHE literature compass for further reading. And Matthias will now continue to show you some examples of PIRCHE evaluations.
Looks good.
Now you can. Perfect. Great. Yeah, thank you, Eric, for introducing the mechanism or what's beyond or what's behind the technique. I'd like to show you now what you can do computationally to use that score in a clinical context. And what I want to show you today is actually what we call PIRCHE ramp, the risk and acceptable mismatch profile, or Tcell epitope risk profile, which is a tool that matches your given patient with a virtual donor population and creates a histogram of the resulting PIRCHE2 distributions. And of course, the better your donor population matches with that virtual population, the better, of course, the conclusions and the more precise the data that you can generate. And what happens technically is basically like this here. In gray, you have all the virtual donors here, and by default, we use 10,500 genotypes created from haplotype frequencies. And in blue, you see here the patient, and each of these virtual donors is then PIRCHE matched with the patient and categorized in different buckets. So basically, histograms created. So this virtual donor here has a rather high PIRCHE score, so we put him into the high-risk bucket, and then we visualize all of that. And this is here an actual screenshot at the bottom of the ramp result page. And to give you a bit more context about the boundaries of these buckets, I also plotted here or added these boxes around, basically referring to the thresholds as were used in the paper from Niels Lachmann in the HAT publication. And you can see here that 75% of these arbitrary donors will be in that intermediate high-risk group for that particular patient. However, you will also find 2 or respectively 8% of the arbitrary donors being located in these low-risk buckets on the left-hand side. And the important thing here to understand is that this is not a fixed image or a fixed plot, but this is individual for every single patient. So it moves around quite a bit, and yeah, it gives you some more context of your data. And to show you what that can actually mean, I have here now two patients, two cases actually. And I created for these two patients now the risk profiles earlier. As you can see, for patient A, the top one, we have a risk profile that's skewed very much to the upper end of the PIRCHE scores, whereas for the patient B, we have quite some donor frequency in the intermediate high risk group. And here I added again these boxes to show you where the previous cutoffs or previous thresholds are. And now we'd like to understand for which of these patients this donor here is the better choice. And we have now donor X, that's the identical donor for both patients here. And we matched him using the PIRCHE method and see, okay, for the first patient, we have a PIRCHE score of 113. So considering these previous thresholds, that's pretty bad situation actually. And for the patient B here, we have a PIRCHE score of 71, which is in the intermediate high risk group. That's okay-ish, could be worse, but also could be better. And the cool thing about that risk profile is now that we can actually use that to individualize thresholds, meaning that we now see for our patient A that the 113 of this donor corresponds actually to a 21% rank, meaning that almost 80% of your arbitrary donors will have a higher score than this particular donor, and vice versa. For the second patient, we see with a 78% rank that only about 20% of your arbitrary donors will have a worse score. Actually, almost 80% will have a better score. So even though the absolute numbers here suggest an okay-ish situation, it might be worth considering this profile here to understand better what's the individual threshold and the individual probability for a patient to receive a well-matched, well-PIRCHE-matched donor. Yes, and with this, I hand it over to Eric again to show you some recent studies.
Looks good.
Good. So in the last part of this webinar, I would like to share some recent data on T-cell epitopes and in relation to memory T cells. As mentioned before, for the B cells, the primary function of immunological memory is to evoke an augmented immune response after reactivation of those cells, after re-encountering the relevant antigen in the body. Generally, this is towards pathogens. But you can imagine that immunological memory may also play a role, for instance, in a second transplantation or in a transplantation that's precluded by a pregnancy or a blood transfusion. And for B cells, there are sensitive Luminex-based assays available to evaluate the presence of pre-transplant donor-specific antibodies and thus get an indication of pre-immunization and the potential presence of B cell memory towards HLA antibodies. For T cells, however, such assays are complicated to execute for two reasons. One is that the frequency of these T cells is pretty low, and two is that they can be directed towards a large spectrum of T cell epitopes presented by various HLA class II proteins. So the presence of memory T cells may explain why individuals with antibodies but no DSA do worse than individuals that have no HLA antibodies at all. That has been shown in various studies. And potentially there is absence of a shared B cell epitope between the immunizing event in the transplantation, but there may still be a shared T cell epitope in those two events. To address this, we use the concept of linked recognition. If there are pre-transplant antibodies, the immunizing HLA should have contained a T cell epitope. And this slide shows you a schematic example, and I will lead you through it. So the recipient has a pre-transplant HLA B57 antibodies and should thus also have HLA B57 specific T cells. The transplanted organ may contain, for instance, an HLA-B60 mismatch, and the recipient does not have pre-transplant antibodies towards the B60 and thus no specific antibodies against this donor. Yet, HLA-B57 and B60 may share T-cell epitopes, and this means that T cells that were raised against the B57 may again recognize a shared T-cell epitope in the HLA-B60 and promote the formation of HLA-B60 antibodies after transplantation. In 2 recent studies, we showed that this approach can work in classifying higher and lower risk transplants based upon the potential presence of shared T-cell epitopes. And the first study here on the left is a collaboration with Toshihide Tomasugi and Takaki Kobayashi. And in this study, the primary endpoint was the development of donor-specific antibodies after transplantation. And clearly, in this graph, the transplant without shared T-cell epitopes, here in blue, perform better than those with shared T-cell epitopes, here in red. And we observed a similar effect when we looked at graft rejection in our Dutch ProCare cohort, here on the right, with a poorer outcome for the transplants with shared T-cell epitopes, an overlap score of more than 0. And in this cohort, we also evaluated the overlap as a continuous parameter. And in the univariate analysis, the overlap score was the only significant factor, and also the multivariate analysis with step forward selection, the PIRCHE2 overlap score was the only factor that was included in the model. And from here on, Matthias will now explain a little bit more about the practical aspects of the PIRCHE2 overlap scores.
All right. Yeah, thank you, Eric, for sharing this recent insights. And thankfully, Toshihide Tomasugi also provided 2 nice examples of his manuscript, which we can show now. And here we have now the first patient, which I want to show you. This recipient's widely immunized against the HLA-B locus after the first kidney transplantation and now received a second kidney transplantation, to which this patient developed the novel DSA against the HLA-DQ locus, and actually rather at the beginning, so only 3 years post-transplantation, so way earlier than hoped for, of course. And we created then this risk and acceptable mismatch profile, also with these acceptable mismatch bars that you can see. The pink bars indicate here the MFIs, and I added on top of that the actual MFI values. And you see the B54.1 and the B39.1 here are pretty strong, strong positive in that reaction. And interestingly, there are no antibody verified B-cell epitopes shared between these bead specificities and the actual donor here. I think there were a few non-antibody verified applets whatsoever shared. Anyhow, it was an antibody against the DQ locus. And what we did next is we created this single antigen bead specificity reaction matrix here, which is a hidden feature, if you want, so of the PIRCHE ramp analysis. And on the x-axis, you see here all the specificities and alleles, and on the y-axis, you see all the peptides that can be derived from these specificities. And if there is a peptide derived from a particular allele, there is a colored box in this heat map, and the MFI here defines the color coding. And I highlighted here now the important sections, and actually the red bars here indicate the positive bead specificities, so the B39.1, B54.1, and in blue you find the class 1 donor specificities. And interestingly, you see here that indeed these positive bead specificities share peptides with the donor specificities. So this might indeed be a potential memory T-cell activation that promoted then antibody formation. And now let's switch to another example, actually a negative case. This recipient was immunized against the HLA-DRB1 locus after a blood transfusion, and then this patient received a living donor kidney from his wife. And you see here again this acceptable mismatch bar chart, this time for DRB1. I added again the single antigen bead assay results here, and you see an antibody against the DRB1 1402 and the 0801. And again, like in the previous case, there were no preformed DSA. Therefore, the transplantation was carried out. Again, no, this time there were indeed shared B-cell epitopes. I think for each of the alleles, there was one B-cell epitope shared, yet it wasn't against no, it was against the DQ locus again, but we see no DSA anyway here. So let's come to this reaction matrix again. This time I added the DRB1 locus here alongside to A and B, and excluded DQ for the moment because it gets a bit small then. And I highlighted again the positive bead specificities here. So again, the DRB1 0801 and 1402, and the donor specificities, so the DR locus and the A and B, just for simplicity here. And if we check out now all the peptides that were introduced by the specificities of these positive beads, then you see that in all of the donor specificities, you do not find a shared peptide. So this can indeed be a no memory T-cell situation. And if this will be validated in one or more studies, this could indeed be a nice thing to keep in mind when defining unacceptable mismatches at time of waiting list registration. So yeah, with these examples, I can just encourage you to go to PIRCHE.com and play around with it yourself and learn about the collaboration with GenDx. And yeah, then back to Andrea and the questions, I guess.
Okay, thank you both for this very nice explanation of PIRCHE epitope matching. So to our audience, if you have any questions for our speakers, please go ahead and type them into the chat box. While we're waiting for questions, I would quickly like to introduce our upcoming webinars. On March 2nd, we will organize a beginner's level case study webinar. For this webinar, we will send you the data of the case study in advance. Our technical support specialist, Elia Lauer, will discuss the results in the live webinar. Also in March, we will have a webinar about the HLA-related MR1 gene in collaboration with Dr. John Trousdale from the University of Cambridge. So go ahead and have a look at our website for more information and to register. Okay, let's take a look at the questions. So, oh boy, we have several. Okay, let's start here. Should this possible T-cell reaction be controlled by the induction therapy that these sensitized patients normally receive before transplantation?
That could be the case, particularly if it targets the T-cell compartment. You may reduce these memory T-cells. That's indeed an option, but it depends on the induction therapy that you use for that.
Okay. How do you account for false positive bead reactivity in the single antigen bead panel?
False positive reactivity to the bead. Well, in our study, we used the the patient's own typing for background checking. So that should be really negative. So there is a background extraction using the Immucore calculations for that.
Okay. We have another one. The PIRCHE 2 score is defined to consider HLA class 2 presentation, but most publications use only DRB1 presentation. Is it possible to predict also DQ-DP presenters?
I think
Yeah,
Matthias can tell more about the technical aspects of DP and DQ.
Yeah, so indeed, I think for 1 or 2 years now, in one of the recent releases, we introduced that option indeed to also predict presentation by DRB345 and DQ and DP. Yet there are not that many studies out there to show the effect. So I encourage you to start that.
Okay. So it seems that these scores are based on HLA allele frequencies in the donor population. So if I am in the US, would I pick a different donor reference population than Europe for calculating the score?
Yeah, so those are actually two separate things here. We use these haplotype frequencies to impute from low resolution to high resolution typings. That's step one. And there, of course, it makes perfect sense to adjust for your regional population. And second is the risk profile, which I showed earlier. This should, of course, also match your local donor population. And what we did is that we provide the opportunity to upload your own local donor population, let's say the last 1,000 donors that you saw in your center, and then you have a very very matched cohort to your local donor cohort. Yeah.
Okay, there was another question about low resolution. Oh yeah, some examples showed low resolution typings, so you're confirming that you can still calculate the PIRCHE scores with lower serological typings. Okay.
Yeah.
Anything
yeah, I can
to
elaborate
add
on
there?
that. So we had a study together with Utrecht and us in Berlin, showing that indeed, if you apply a multiple imputation, you have, of course, certain noise that you introduce by that method. However, that noise is below one risk category, if you want so, for I think 99% of the patients. So it was rather fine for the retrospective studies, and in the future we may go for NGS maybe.
Yeah, and what is important to know is that if you use NGS for your recipient population, you significantly increase the preciseness of your predictions. So whereas it will be difficult to type all your donors in donor procedures in time with NGS typing techniques, it is feasible for your recipient. So yeah, that's certainly advised to deliver typings for your recipients at the highest level possible, because it will reduce the noise in all the analyses that that we showed.
Okay. So very interesting case studies, thank you. In PIRCHE, how can we view the single antigen bead specificities table?
So if you created this risk and acceptable mismatch profile on the result page, there's actually a button called Load MFI. And with this button, you can select reports generated by 1Lambda Fusion 4.3 and 4.4. So there are dedicated report files that can be read by the PIRCHE website. And Immucore also just released an interface where you can export your antibody test results directly to the PIRCHE website, and then you get these additional bars and this matrix.
Okay. Let's see, I think we have time for a couple more. Do you know anything about the outcomes of epitope mismatch in liver transplantation?
Well, that's an interesting one. There's a publication on PIRCHE in liver transplantation showing some associations. Generally, there's quite some discussion of HLA matching and mismatching and the effect thereof in liver transplantation. So well, as far as I think, this is also dependent on the transplant strategy, but there are publications showing that PIRCHE can indeed, well, is associated with the outcome of liver transplantation. I think it was also
Eric, you had one publication, you showed one publication earlier and cited that from Magdalena Messaros.
Yes.
And this is actually pretty interesting because it also takes into account the immunosuppressive therapy, so the CNI levels of these patients. And the long story short, it was like, if you have low-risk patients, you can withdraw CNI, but if you have high-risk patients in terms of epitope match grades, then you shouldn't withdraw CNI because these patients had way more graft failures than the low-risk or low-PIRCHE patients. So that's indeed a good read.
Okay. Okay, last, and this one's a little bit more technical. How do you upload the typing data to PIRCHE?
Yeah, so there are indeed various options, some of which are more on the manual side, but no one wants to type in his typing data manually, so that doesn't make too much sense. Then there are wizards on the PIRCHE website to load GL strings or HML data, and most of the current vendors also have a link to PIRCHE, like for example in NGS Engine. You also have this cool send to PIRCHE functionality. If you right click on your sample, then you can directly upload your typing data.
Okay, great. Well, I think that is all the time we have today. So I just want to once again thank you both for being guest speakers during a GenDx webinar. This was very informative for me especially. And also thank you to our audience for attending. If you have any further questions that we didn't get to or that you're still contemplating, go ahead and send those to us at education@gendx.com and we can share those questions with our speakers and they can help you further. So I wish you all a good day. Bye.
Bye-bye.
Bye-bye.